are some lymphocytes, and this is kind of going off on a tangent a little bit, but I'll just mention there are some lymphocytes that would have host specificity that would enter the bloodstream that could recognize antigens or proteins that are common to host tissues, and therefore, if activated appropriately, would respond against host tissues in the same manner that it would respond against the recognition of the pathogen. And so that would cause an autoimmune disease, like lupus or diabetes, that would result from the damage to healthy tissues by self-reactive lymphocytes. But under normal circumstances, and because of the types of activation events that take place in the body, it's very rare that those lymphocytes would ever become activated, those that do escape this screening process. But the majority of lymphocytes that possess self-reactivity will be screened and culled from the population before these cells are allowed to complete their developmental program and then be exported into the bloodstream. So the body will maintain millions of lymphocytes. That's due to the daily production of B lymphocytes from the bone marrow and T lymphocytes from the thymus. The body will maintain a homeostatic level or number of lymphocytes, each with their own individual antigen specificity, such that we'll have a bank of lymphocytes available that we can make an adaptive immune response to any pathogen that we could ever encounter. So that's the goal of the adaptive immune system. It's activation of lymphocytes that recognize specific features of a particular pathogen and can focus defense mechanisms onto that pathogen. So where does the adaptive immune response take place? The innate immune response takes place at the site of injury, when a microbial invader has been introduced because of injury to protective epithelial linings. Defense mechanisms are focused directly onto the site of injury, where the microbial invader will be recognized and hopefully eliminated and prevented from establishing the site of infection. However, let me go back a couple of slides just to show this bottom figure here. I just went back a couple of slides. However, if the innate immune system is overwhelmed, then we need to bring together lymphocytes and the pathogen. Because remember, we've got a very large population of lymphocytes. We've got millions of lymphocytes, each with their own individual antigen specificity. Therefore, for any one particular pathogen, the frequency of lymphocytes that could recognize that pathogen has been estimated to be 1 in about 10,000 to 1 in about 100,000. So out of a population of lymphocytes, that's essentially a few needles in a haystack that would have an antigen receptor that would recognize a specific antigen elaborated by that pathogen. And so therefore, it's important to select those lymphocytes from the population to be activated and induced to proliferate and differentiate to display their effector function by focusing a particular defense mechanism onto the pathogen itself. So how do we bring the two together? Right, throughout the body, we have specialized lymphoid tissues that can trap pathogens. They're strategically located throughout the body where pathogens enter the body. And therefore, pathogens can be trapped in that manner, as well as antigen-presenting cells are found along with macrophages underneath the protective epithelial barriers that can acquire the pathogen and directly migrate to the nearest specialized lymphoid tissue and make antigens available. So antigens can be trapped as they're carried and filtered in secondary in these specialized lymphoid tissues, and also antigen-presenting cells from the site of infection can migrate and enter a secondary lymphoid tissue. So these tissues are strategically located where pathogens normally enter the body, through the skin. And if they enter through the skin from an injury, two things could result from an establishment infection. They could enter lymphatic capillaries and then be carried to lymphatic vessels, which would then converge into a lymph node, which is a secondary lymphoid tissue where the pathogen can be trapped. So lymph nodes filter the lymphatics, and that would be due to pathogens that enter tissues that gain access to the lymph lymphatics. On the other hand, pathogens can enter the bloodstream. And so the blood is filtered by the spleen. There are antigen-presenting cells in the spleen that will recognize and trap the pathogen there, as well as gut-associated lymphoid tissues. Those microorganisms that enter through the respiratory epithelial linings or the gut linings, they can be trapped by specialized lymphoid tissues that are found throughout these areas, underneath the internal epithelial linings of the body. So there's three specialized types of lymphoid tissues that are collectively called secondary lymphoid tissues. And they essentially trap, number one, they have two functions. One is to trap pathogens when they enter the body. And therefore, they're strategically located to all parts of the body where the pathogens normally would enter in a natural infection. And then secondly, what takes place is that lymphocytes that circulate throughout the body, these would be naive lymphocytes, meaning that they have yet to recognize an antigen and be activated. They just essentially have a surveillance function at this point in their lifespan. Naive lymphocytes will visit each secondary lymphoid tissue as they travel throughout the body. The reason being is that these lymphocytes, that's where they acquire their growth factors to maintain their lifespan. So all these somatic cells have a certain lifespan and require either contact with other cells like the basement membrane, for example, or exposure to certain hormones or growth factors in order to maintain their viability for their lifespan. So lymphocytes, like all other cells in the body, require growth factors. And therefore, they acquire these growth factors from secondary lymphoid tissues. Therefore, that induces them to enter these tissues. And therefore, this provides a meeting place that if we've had an infection in the skin, and then the microorganism has established a focus of infection, and bacterial cells have either entered lymphatic capillaries and entered lymphatic vessels, they'll be trapped once those lymphatic vessels converge into a lymph node, or antigen-presenting cells in an infected area can enter the lymphatic, can engulf the pathogen and enter the lymphatics and travel to that particular lymph node, just as an example. And so in this lymph node, it will swell up in that a large number of lymphocytes in the circulation will be entering the lymph node that day, seeking survival factors. And so that ensures that this 1 out of 10,000 to 1 out of 100,000 valuable naive lymphocytes or valuable candidates that can recognize that specific pathogen will be brought together to the pathogen. So lymphocytes address the problem secondary lymphoid tissues address the problem is that if you have a large repertoire of lymphocytes, millions of lymphocytes, roughly each with its own antigen specificity, how is it possible to activate an adaptive immune response before disease kills the animal? Well, secondary lymphoid tissues provide the solution to that problem because they trap pathogens wherever pathogens enter, in the places that pathogens commonly enter the body, and then they recruit lymphocytes from the bloodstream to migrate into those secondary lymphoid tissues, and therefore the two are brought together because a very large number of lymphocytes will be migrating through these secondary lymphoid tissues that day on the day of the infection. So let's finish up and look at some of the characteristics of lymphocytes. So let's say what takes place in the secondary lymphoid tissue. Those that serve the upper respiratory tract, for example, or the gut, or the spleen that filters the blood from blood-borne particulate matter, which would include pathogens, as well as lymph nodes that filter the lymphatics that drain from tissues that would filter pathogens. So let's say we have an infection that's become established, and therefore the pathogen has been trapped in a secondary lymphoid tissue. And lymphocytes are entering the secondary lymphoid tissue, seeking growth factors. And therefore the two are brought together. Antigens are made available by antigen-presenting cells for recognition by lymphocytes. And so this would allow the activation of those lymphocytes that would be most appropriate for the infection at hand. And so we have cell-mediated immune response and humoral-mediated immune response based on how the pathogen replicates and what type of disease-causing characteristics that pathogen possesses that initially allowed it to overwhelm the innate immune system and establish an infection and establish itself as a dangerous microorganism. So some pathogens, like viruses, for example, can replicate inside of cells and come in and commandeer the host macromolecular synthesis machinery to replicate their nucleic acids and then direct the expression of viral proteins. So some pathogens come in and commandeer cells in which to replicate themselves. And so the problem here is that a silent viral replication that's taking place inside the cell is very difficult to be recognized by the immune system. And so this infection will be allowed to spread as these viruses bud out of cells, or it depends on the virus. If the cell lyses, when virions leave an infected cell, they will bind to receptors on neighboring healthy cells and initiate another round of infection, and the viral infection will spread. So the adaptive immune system, cell-mediated immunity, is dedicated to how this particular pathogen replicates. And therefore, one arm of the cell-mediated immune system is that naive cytotoxic T cells, so-called cytotoxic, because once they've been activated in a secondary lymphoid tissue and receive the appropriate activation signals as well as the antigen from antigen-presenting cells, those T cells with cytotoxic potential will proliferate and then differentiate into effector cytotoxic T cells, which means they now possess characteristics to eliminate the pathogen from the body. And the particular characteristics here would be that the cytotoxic T cell would differentiate between healthy and infected cells and induce infected cells to undergo apoptosis. So they would recognize and eliminate, or recognize and kill infected cells to stop viral replication. Now, viruses outside of cells that are leaving an infected cell and are being carried, let's say, in the interstitial fluid to healthy cells in that tissue to reinitiate infection, that can also be addressed by another arm of the immune system. But right now, viruses that are a silent replication that's taking place inside a cell can be recognized by cytotoxic T cells that are specific for features peculiar to that virus, and therefore mediate the specific recognition and elimination of infected cells to break the viral life cycle at that point. And then there's another arm of the immune system that can address virions that have left infected cells that are going to infect their healthy neighbors. And so once these cells have been induced to undergo apoptosis, then tissue macrophages, which also have a scavenger function, will recognize and remove apoptotic cells. Now, other T cells that mediate cell-mediated immunity have two different functions. First of all, they can activate those cells, other cells in the immune system that are responding to the same pathogen. So in this particular case, this would be a pathogen that replicates extracellularly and is producing a toxin. And so they're so-called helper T cells because they provide the appropriate activation signals. So on one hand, there's a class of helper T cells that are activated in secondary lymphoid tissues to proliferate and then differentiate, and then migrate from that secondary lymphoid tissue, enter the bloodstream, be carried back to the site of infection, and induce macrophages that have engulfed pathogens that are resisting being killed. So pathogens can antagonize or resist phagocytosis, and if they're phagocytized